Malhotra, the Global Chief Technology Officer at Cipla. To you, Ms. Geena.
Thank you, Navneet. And hello and good evening, everyone. I'd like to thank the IP management team for inviting me on this platform to speak on a topic that has rapidly gained attention and importance over the last decade. And this is the pharmaceutical development and manufacturing under QBD framework. Albeit not new to the manufacturing industry, the usage of QBD principles has been embedded in automobile and other industrial companies for over 6 decades now. And adoption in pharma has been relatively young since the concept was introduced in the mid-1990s. Now, over time, the quality by design approach was introduced in the pharmaceutical industry, and it has evolved rapidly with the publication of 3 guidances by the International Council of Harmonization, namely ICH Q10, Q9, and Q8. And these guidelines describe the elements of pharmaceutical development, the quality risk management, and the pharmaceutical quality systems. So I would really encourage that all of you read these many times over, because this is really, it brings in the essence of everything that we want to talk about today. So if you go to the next slide, please. So ICH-QH defines QBD as a systematic approach to development that begins with predefined objectives and emphasizes product and process understanding and process control based on sound science and quality risk management. So this is a very clear and easy to understand description, at least in theory. In its form, QBD can be explained as an orderly, well-planned procedure to assemble and deliver quality. So if you go to the next slide. So historically, you see the slides in 2 parts. There's a left part that talks about quality by testing, and the second is the topic of discussions, which is quality by design. So historically, quality by testing has been utilized to develop processes. However, there have been many challenges. Product quality and performance is supported by the end product testing, and it leads to limited science-based understanding of the process, managing of key variables and their effect on end product quality. And it also has a limited understanding of the process design space and relationship with the product quality. So in general, there is less time devoted to understanding why processes fail, and there's more emphasis on testing and reworking out of specs product as necessary. And it also ends up results in data intensive submission, disjointed information, without really considering the entire process, and the specifications are based really on batch history. And now when you look at the current approach of QBD and the topic of discussion today, quality is built into the product and process by design based on a scientific understanding. And the specifications are based on product performance requirements. So there are many advantages of quality by design. The first is that it is right first time, reduced costs and less process downtime. There's a science-based understanding on the process results in minimized batch failures or rework. There's reduced time to market for new drugs. And process changes within the approved design space is permitted without regulatory resubmission. Also, with the use of patch tools, real-time process monitoring and trending reduces the analysis burden and improves the product quality. So using these QBD tools like DOE and PAT, the variable inputs that we have, in spite of the variable inputs, we can actually achieve constant outputs. So then the question is that with all of these advantages, why is there such an inertia to adopt quality by design? And I would think that one is that all stakeholders must have agreement. The question is there a corporate inertia? Is there truly a management buy-in? And do consider the initial cost of equipment and training and developing quality by design-oriented information systems to capture necessary documentation. Often there's a lack of understanding of the actual cost savings and the positive business model. And so using QBD, we will actually see that there will be a paradigm shift from the usual approach of development, and this would represent a cultural change that must be addressed with the change management techniques. And the importance of change management is not recognized by many organizations that deploy, whether it's QBD or whether it's any other improvement techniques. And as a result, the promise of the associated change is seldom realized even in minor terms. So if you go to the next slide. So the question is, how to go from definitions and guidelines to the actual process and product development in the real world situation. And the uncertainty in the answer drives many companies away from QBD to adhere to the more traditional approach of pharmaceutical development. So this slide represents a very simplified workflow with all the important elements that must be present in QBD development of a pharmaceutical product. A lack of understanding of QBD in its entirety is the large stumbling block for its use. And as we've heard before, that for a successful strategy in any endeavor, it is best to begin with the end in mind. And so the first step is to understand the critical outputs of QBD and then to identify the critical building blocks of QBD. Like, for example, improving your process understanding and to control or reduce the risks. So the outputs of design space, the process control procedure, the risk level, whether it's qualitative or quantitative risk assessments, these are consistent with this approach. And before the results can be realized, however, the building blocks of QBD need to be assembled. So one of the first elements to be defined is the quality target profile, the QTPP as we know. It's a summary of the desirable characteristics a product must have to ensure the desired quality, taking into account the safety and efficacy of the drug product to the patient. The second is to identify the critical quality attributes, characterize all the input raw materials or other inputs and the variations of the input raw materials, identify the critical process parameters, characterize the design space, ensure process capability and analytical method capability, control and robustness, and create a process model monitoring and maintenance. Now, the multidimensional combination and interaction of the process inputs that have demonstrated to maintain the CQAs within the specifications is called the design space. And we often hear about what the design space is. The concept brings about a certain flexibility to the table, since alterations made within the design space are not considered major changes. And the combination of risk assessment and the data analysis is one of the pillars of QBD, and the opportunities of acquiring and managing the data are pivotal for a successful pharmaceutical development and life cycle management. So if you go to the next slide, I would like to elucidate this in the simplest case study. So this is a case study for developing extended release tablets using QBD approach. The CQAs were identified and the targets were set as per requirement. And in this case, the most important CQA was identified as assay and dissolution. Going to the next slide. So the materials of formulation variables impacting these CQAs were considered here. The impact of variables on the CQAs were rated high and low based on the severity, and the high risks are colored in red, and the variables having high risks of CQAs were considered further. So going to the next slide. Yeah. So preliminary trials were taken to understand the role of each of these high-risk excipients, and we fixed a few of them. And finally, we identified 3 variables that were selected for evaluations, which was the API particle size, then there was a percentage of the release modifier, since it's an extended release product, and then there was the concentration of the stabilizer that was used. Going to the next slide, please. So based on the variables criticality and availability, the I-optimal design was selected. Now, I-optimality minimizes the average prediction variance of your model over a region of measurement parameters So it is more naturally applied when you know the form of the model you want and you want a good prediction over your design. And for this reason, I normally finds more use in response surface optimization context. Now, typically, in the design of experiments for estimating statistical models, optimal designs allow parameters to be estimated without bias and with a minimum variance. A non-optimal design requires a larger number of experimental runs to estimate the parameters with the same precision as an optimal design. And in practical terms, optimal experiments can actually reduce the cost of experimentation. So if you go to the next slide, please. The next slide, or is my video slow? Please go to the next slide. Yeah, thank you. So this slide shows some of the commonly used DOE designs. Now, a strategically planned and executed experiment may provide a lot of information about the effect on a response variable due to one or more factors. Now, many experiments involve holding certain factors constant and altering the levels of another variable. And this is the one factor at a time, OFAT approach, as we know. But however, this is inefficient when compared to changing factor levels simultaneously. Now, DOE is a powerful data collection and analysis tool that can be used in a variety of experimental situations, and it allows for multiple input factors to be manipulated, determining the effect on the desired output or the desired response. Now, by manipulating the multiple inputs at the same time, DOE can identify important interactions that may be missed when experimenting one factor at a time. And all possible combinations can be investigated, whether it's a full factorial or only a portion of possible combinations in a fractional factorial. So go to the next slide, please. So I don't necessarily want to discuss these plots here, but this is really an opportunity for you to see the kind of data and output you get to analyze further. And this is also really for you and the management to appreciate the kind of skill sets you need to acquire, nourish, and allow to bloom in your organization. This is really setting up a very elite set of scientists that will do this work. So coming back to this slide, these are contour graphs of showing how the two variables that we picked up, namely the release modifier and the stabilizer concentrations, how they impact the dissolution response when there are two different particle size of API dissolution, a low particle size and high particle size. And what we see is that the lower level of A and B show a higher dissolution compared to the higher levels. And the arrow indicates the region in which we need to consider to achieve the target for dissolution, where the dissolution target was required to be more than 90% at 24 hours. The next slide, please. So these are the contours of all the responses were overlaid with target specifications, and the design space was established as a yellow region. And this is a space where we will meet our target. So we saw that there was more flexibility that could be achieved with a coarser API as the design space was larger. And therefore, the question for us is, where do we operate within this design space? Going next slide, please. So to get more confidence, we considered 95% confidence interval of the design space. And the key point is, where do we play within this so that we are not on the edge of the operations? And if we select the point on the edge, then what will be our chances of failure? Next slide, please. Yeah. So here we see that when we looked at the concentrations of the stabilizer, when the concentration was at 4%, there was a probability of failure at 60.7. When the concentration changed to 5%, the probability of failure reduced to 5%. And when it was at 5.5, the probability failure is 0.87. So what I'm trying to just explain to you is that using these tools, you are able to identify the selected conditions within the design space where the failure would be less or very low. And hence we selected a concentration of 6% for the stabilizer. Next slide, please. And And then finally, we had a final optimized formula for the two factors A and B at 12 and 13% using this tool, where we found that even a coarser material could be used where failures of chances were negligible. So next slide. So at this point, we know the variability of the process parameter and the design space. And from the DOE, we can get multiple number of outputs, and we can actually create using Monte Carlo more than 100,000 simulations, and that can help you calculate the process capability. Now, this is to understand how variability in process parameters can impact the performance of a CQA. And Monte Carlo simulation is one of the best tools to establish the robustness of the process without performing any experiments. So considering this, if you look at the histogram at the right end, we see that when there's a standard deviation of 1% for the solid content, the CPK of the critical quality attributes is 1.2, as against desired greater than 1.33. So we can identify the CPPs that will allow for a standard deviation which is below 1% to maximize the process capability, and then accordingly define the control strategy. Going to the next slide, please. So I'd like to take you through some investments in the PAT tools that have certainly helped drive real-time understanding of the impact of process parameters and helped design, analyze, and control manufacturing through timely measurements of critical quality and performance attributes of raw materials and in-process materials. And these PAT tools help to define design space via multivariate data analysis encompassing the critical processing parameters. It also helps confirm that the process is operating within the design space via monitoring and trending. And you can use the PAT tools to support validation efforts in process design, qualification, and verification stages. Next slide. So I'm not sure if you are able to click on the right side of this slide. There's a little small video. This is an NIR PAT tool used for BU monitoring in an octagonal blender. I mean, if you can click on it, otherwise we can move. Next slide, please. Yeah, and here each component shows a unique spectra of the different intensity and positions for their bands based on the individual excipients and the actives that were put in this blender. Next slide, please. So to extract the relevant information from raw spectra, we need to give certain mathematical treatment to the spectra, and this will enhance and sharpen the position of the peaks and reduce the effect of the spectral noise and reduce the effect of instrumental noise. Overlapping of bands will be minimized. And in this case, the standard normal variate is a normalization method. It corrects for the spectral difference, and derivatization helps resolving the peaks and sharpens the peaks. So next slide. Yeah, so this slide shows the spectral variation of blend homogeneity. And at the initial stages, you can see you've got the 4 quadrants. So if you look at the top left quadrant, these are the initial stages of blending in 2 minutes, where you can see that the spectral variation is high. And with time, the variation is reduced. And there's a minimum variation at the end of blending when you look at the spectra in the right bottom corner, where we see that the blending is optimized between 12 to 15 minutes. And so these are the 2 approaches where you can get blend homogeneity data, both, and this is a qualitative data. You can get both qualitative as well as quantitative data, but this particular one is a qualitative method to demonstrate the changes in the spectral variation. Next slide, please. So this is another qualitative data, and where we've used the moving block standard deviation method. In this, MBSD is the moving standard deviation of the input signal along with each channel independently over time. Next slide, please. And when we want to build a quantitative analysis, a graded response is required for preparing a calibration model. And for this, we have to use different concentrations of the blend. We create a spectra with different concentrations and check if we get a gradient and this helps us quantify the changes of the performance. Next slide, please. Yeah. So this is again a quantitative method to determine the BU using inline NIR PAT2. And the NIR based prediction is seen here, which is, as you can see, this is very close to 100% and closely matching the reference analysis of sampling at the end and doing the HPLC, doing it by comparing it with the HPLC results as well at the bottom of the slide. And once the model is developed, the model is then applied for monitoring the BU as this plot shows it. Next slide, please. So this slide demonstrates another emerging technology in pharma, that is to use CFD, computational fluid dynamics. You could use this for solids, liquids, and semi-solid suspensions. In this instance, the CFD has been conducted to understand the mixing profile and the shear at different vessels with different types of agitators. And this will help to determine and establish any scale-up of batch size or equipment through ensuring homogeneity of suspension at different scales and batch size. Next slide. So I'd like to leave you all with a very, very progressive and real doable approach, which is the next level of QBD. It's this next trajectory will take us into an orbit of in silico design space using in silico modeling, discrete element methods, CFD, and digital twins. And we've been using different modeling and simulation tools across product life cycle. So in the next few slides, we will show you examples of application of modeling and simulation tools for pharmaceutical development. And this could be used for scale-ups, for commercial manufacturing, and continual improvement. The next slide. Yes, so this is an in silico model for developing a design space. Now, for manufacturing of tablets, for example, it is important to have a good balance between flowability and compressibility. You're all very familiar with it. So while deciding on a new formulation, and selecting excipients, it will be useful to know whether a given formulation will flow and will have the required compatibility to be compressed into tablets that can handle the manufacturing and shipping stresses. So this model helps in creating in silico design space for any given formulation without doing any experiments. And once we have and for this we need to have the material properties like individual excipients and API, like the bonding capacity, the tensile strength at zero porosity, and the complete design space around it can be predicted. So in silico design space can be used for screening formulations and identifying the right solid fraction for example, in roll compacted ribbons and in compressed tablets. Next slide. This is a DEM, discrete element method. It's a physics-based modeling technique to simulate bulk powders flow during blending and hopper flow, etc. Now on the left, again there's a small video, and I think this doesn't work, but it's a DEM simulator. And to start the simulation, a model of equipment is created, and then the particles present in the formulations are generated into the equipment with material properties like particle-to-particle friction coefficient or particle-to-wall friction coefficient to simulate the bulk flow of real material. The equipment model is in the simulator is then given the required RPM as per the real equipment, and the model keeps track of all the particles and forces acting on them and gives the motion as per Newton's second law. And throughout simulation, for every rotation, we get a mixing index, which is an indicator of mixing. In similar way, this tool can be used for even a new process design or scale up to identify the right equipment and process parameter for robust blending. Next slide, please. So this is an adaptive manufacturing using digital twins. Now, a major challenge that is faced in manufacturing is changes in the input material properties. And in order to enable consistent downstream product quality, the manufacturing process needs to be adaptive. And digital twins help making this process adaptive. For example, for building a roll compactor digital twin, the input material needs to be characterized for its frictional properties, and then with a few short calibration runs, the digital twin is calibrated, and using a calibrated digital twin, the complete design space can be predicted for a product on the machine, and the variability of the input material can be handled by making the process adaptive to get consistent relative ribbon density as in the instance of a roll compactor. And similar approaches could be used for compression machines as well. Next slide. This slide is about using advanced analytics to build empirical model. So pharmaceutical manufacturing processes generate a wealth of data which, if rightly used, can be used for continual improvement of process performance, ensuring process robustness. So the graph on the left shows a batch fingerprint which uses advanced analytics to build empirical models using possible variables like material properties, process parameters, etc., and projects a multi-dimensional space into the 2D space. And a good model is shown in the graph on the left. It segregates the 2D space into a high performance and a low performance region. And you can use this for identifying the optimal process settings to move the process to a higher performance region. Go to the next slide, please. So as I conclude today, as we've seen from other industries and more recently in the pharmaceutical industry, QBD is an effective method for developing new products and process and enables effective technology transfer and the optimization and improvement of existing processes. QBD looks set to become an increasingly important one for pharmaceutical industry as more and more companies are looking to boost production output, reduce throughput times, and lower costs by shifting from batch product production to continuous manufacturing. It also works because it fosters process understanding that is fundamental to the creation of the design and control spaces and to sustain performance. This process understanding enables the reduction of process variation and the creation of process control systems that are based on sound science and quality risk management systems. And the implementation of PAT, modeling, and simulation should be considered case by case. And additionally, I see today that FDA and EMA and other regulator bodies further scrutinize production process to ensure the highest level of safety, and QBD will make it easier for companies to demonstrate that they are operating within acceptable limits. As regulatory authorities start to insist that companies have a design quality into their products at every stage of the pharmaceutical value chain, the use of QBD will become ever more important. And lastly, invest in technology. It will pay off in the reduction of waste, loss of batches, reduction in downtime, optimizing resources of plant, material, and machinery, and a caution to my pharma fraternity. For too long, we've used processes and equipment that were fundamentally designed many decades ago, with incremental changes decade after decade. And these don't really support to a large degree the intelligence, automation, and digitization. So for long-term value-backed vision, I would ask to embrace technology and equipment that would support this. Thank you, and thank you for listening today.